Hello, welcome to my coronavirus classroom. I'm Janessa Jacobs, and this is Skeletal Muscle Tissue Part 2. This is the third time I've tried to record this particular lecture. Actually, it's like the fifth time, but it's like third time for Part 2, so hopefully third try is the charm. If not, we may just rely on old YouTube clips and a little bit of a mashup of everything that wasn't covered in those at the end. We'll see. Hopefully it'll go. Right now my camera is showing me this little red circle, which leads me to believe that it is recording. But this is what happened yesterday. And I went through this whole big, beautiful description of things and heard a beep after 15, 20 minutes. And at that point, my camera was like, oh, I just turned off due to low battery, even though there was plenty of battery and whatever. The point is that here we go with one of my favorite topics of anatomy and physiology, the sliding filament model of contraction. As you should know by this point in the semester, I love muscles and this is how they contract. So I really hope that this is a true red recording circle. I don't have an actual cameraman, it's just Alex, who's, you know, the plastic model, and Shaniqua, who's that model, and Kelly, who's that model, and Marcus, who's that model, and none of them are telling me if this camera is actually recording. I'm relying on a little red dot that says right now it's recording. So here we go. Here's cheers to hoping the sliding filament model of contraction lecture actually goes as it's supposed to. And the reason I'm so angry about it is because yesterday I had this beautiful picture up of my thick filaments and my thin filaments, and it was different than this, I'm realizing now. So, okay, here's my thick filament with my myosin protein. This is my M line, which means that this is the middle of my sarcomere. And pointing out from the middle of my sarcomere to the ends of my sarcomere are all of these myosin proteins. And I can only draw on my little whiteboard here, so I can't show you how they're actually wrapping around this cylindrical structure, the myofibril, in 360 degrees. But that's what's happening in your muscle fibers, okay? On my board, it's just these little double-headed myosin proteins that we find in thick filaments. So if this is the M line, and that is the middle of my sarcomere, what is going to be my end boundary on both sides? You should know this by now. It's the Z-disc, right? A sarcomere is the smallest unit of contraction that goes from Z-disc to Z-disc. So on one side of this sarcomere, I have a Z-disc. And on the other side of the sarcomere, I have a Z-disc that I'm not going to draw on my beautiful muscle figure over here. So, pointing away from the middle of the sarcomere, I have the rest of my thick filament, or for this particular portion, which is going over here. Okay, M line. This whole thing is a thick filament. Hold on, my love. This here is my Z-disc. And what my Z-disc is doing is it's anchoring my thin filaments, which have that globular actin protein, and this is horribly drawn on the drop of a dime, sarcomere here, and now here comes my 4-year-old. So we're just gonna pretend this looks exactly like what it's supposed to. Okay, so this is a thin filament. This is a thin filament. Here's a thick filament. I've got another thick filament right here with all these myosin proteins sticking out all over the place. Why? To grab on all the thin filaments that are in here. There's a thick filament. And what I have right here is this spot called the H zone, and that is free of actin. And so if I see the H zone disappear, I know that I have slid this actin filament completely over this myosin filament, and now I've got a shortened sarcomere and a contracted muscle. This whole thing is a filament model of contraction because these things are microscopic. And so we know what's in them, and we can see what a relaxed sarcomere looks like, and then what a contracted sarcomere looks like. And one of the things that we'll notice in a contracted sarcomere is that you don't have an H zone anymore, because there's no spot that's free of actin. And that's because our thick filaments are going to slide these thin filaments over themselves. They're not changing their location. They're just moving these little myosin heads. And so the thin filaments are going to change their location because they have no choice. My myosin heads are going to pull them along, and my thin filaments are going to come over, and my H zone, which is free of actin, is going to disappear because there's not going to be a spot that's free of actin. As these guys who don't change, what is it? They don't change, our thick filaments don't change width? I mean, they're moving. I can't say they're not moving because these myosin heads are pivoting and things that we're going to talk about in a minute. But this length here, this distance isn't changing. What is changing is the span from Z disc to Z disc, as these guys who are just sitting here pulling these thin filaments pull this thin filament, pull that Z disc over, and they pull this Z disc over, and now all of a sudden I contract my sarcomere. I shorten every sarcomere in the muscle fiber, and that's where we're going next. And then as I recruit more and more muscle fibers, I'm able to do various types of contractions to allow me to do various types of work. So the sliding filament model of contraction is this model that we have to explain what we observe in a relaxed and a contracted sarcomere. That was a whole bunch of bare contraction of sarcomeres to do lots of jumps right there. Thank you, little man. So the model is that we have these thin filaments slide over our thick filaments, and in that shortening of every sarcomere along the length of a muscle fiber, we're contracting the muscle fiber and generating tension. So what we could say for the rest of this slide, I don't think I want to go through the specific mechanisms of the cross-bridge cycle right now because I don't think I have to. Yeah, we have a slide for that later. So for our sliding filament model of contraction, we could say this explains the mechanism by which thick filaments slide thin filaments toward each other to shorten the sarcomere, contract the fiber, and generate tension. Explains the mechanism by which thick filaments slide thin filaments over themselves. Sure. To contract a fiber. What kind of fiber? A muscle fiber. And generate tension. All right, so muscle fiber contraction. If I recall, the copyrighted figure that I'm not copyright infringing that would be here right now is a table that sums up how we can get a muscle fiber to contract by breaking it down into 3 anatomical structures. And so muscle fiber contraction really can be focused in on and broken down to like these 3 different areas that are involved anatomically. And there's a lot going on physiologically, so that's where it kind of gets confusing. And this is why I actually like to start the semester with the nervous system, because the first area that's involved in muscle fiber contraction is the neuromuscular junction. Neuromuscular junction. This is where our lower motor neuron that you thankfully have already learned about is going to meet our muscle that you're learning about right now. So instead of having you suspend your disbelief about what's happening in this hugely important first step, you already know what's happening in this hugely important first step, which is the events at the neuromuscular junction. And I'm writing it up this way because I want you to be aware that in muscle fiber contraction, as complicated as it may seem, there are only 3 parts of the muscle, or well, of the whole system involved. And yes, each part has these complicated steps. Whew, but that's okay, because today you can master one part. And tomorrow, you can master the next part. And the next day, you can master the next part. And then the next day after that, you put it all together. How do these 3 parts work together to contract a muscle fiber? And then you send me an email with your 3-part description of muscle fiber contraction, and you say, is this correct? And I look at it, and I say, oh yeah, you mostly got it. Let's look at this one part. And then instead of telling you how the truth of it things, because I'm telling you that right now, I'll ask you questions. Well, think about it this way, or think about it this way. And then what happens is you can arrive at the answer yourself because you understand it. And that is where we're trying to get your understanding, okay? So don't be afraid. Muscle fiber contraction can be broken into 3 anatomical areas. First, the neuromuscular junction, where our lower motor neuron makes contact with our muscle. Then, the sarcolemma and T-tubules of our muscle fibers, which we already met. And then, the sarcomere that we've also already met. And so sure, we're going to dive in and get some greater details in each of these spots, but that's why we had the last lecture, was to lay this foundation so that now you could look at each of these like little areas that have this hugely complex physiology and not be intimidated, because it's actually not that bad. You have all these glorious skeletal muscles that you can contract. You have this beautiful brain that can learn. And I know for a fact, 100%, because I did it, that human brains can learn this. And you got it, okay? So, muscle fiber contraction begins at the neuromuscular junction. And let's see. Yeah, we're going to talk about the nitty-gritty details next. So the figure in your book that I'm not copyright infringing, but I'm remembering because I got a great brain like you do. So it describes all of the events of an action potential coming down the lower motor neuron, and calcium enters the synaptic knob, causing exocytosis of acetylcholine. Acetylcholine will then cross the synaptic cleft and bind to the muscle fiber on this spot called the motor end plate. And I know that with your book you get these videos called A&P flicks. So you could log on to the website that you've already paid for and watch all their fancy videos. You could also just like YouTube search neuromuscular junction. All right, so the second event after stimulation at the neuromuscular junction is going to occur as we have this excitation of our skeletal muscle that will be coupled to the actual contraction. So the second step in muscle fiber contraction is what's called excitation, contraction, coupling, which I will from now on abbreviate as ECC, and the anatomical structure or location that this is occurring is what's called the motor end plate. It's where a lower motor neuron that you've already learned about makes contact with the muscle fiber, which is what we're learning about right now. So our neuromuscular junction is that. And then what's going to happen right there is that we're going to excite the muscle. And then now in excitation contraction coupling, the structures that are involved are the sarcolemma and the what? T-tubules, right? So these are going to be able to conduct action potentials deep into the muscle fiber around the myofibrils. So for muscle fiber contraction to occur, the first anatomical location is the neuromuscular junction. And what we have there is excitation of a muscle fiber at this spot called the motor end plate on the muscle fiber that we're going to talk about in a minute. So we will excite there, and then, so excite. What we're trying to do is contract a muscle. So the events in excitation contraction coupling are the ways that we conduct this action potential that's generated here down the sarcolemma and T-tubules so that we can lead to contraction. And then the third event is the cross-bridge cycle. And the cross-bridge cycle is happening at the anatomical location we just met, the sarcomere. So this is at the sarcomere, and this is going to be the sequence of events that occurs when calcium, which is released in the excitation contraction coupling phase, binds to the troponin tropomyosin complex we talked about last time, moves the regulatory protein, exposing those myofibrils, the myosin head binding sites on our thin myofilaments, to exposing the myosin head binding sites so that on our thin filaments, so that our thick filaments can reach up and grab them, and we will actually have that shortening of muscle. So there's a summary figure in your book. I am like 99 to 100% sure of it, which is what would have been up here in a normal class, and it's taking you through all of the steps in these 3 steps. Okay, so we're going to look at these areas in more detail. Let's now look at what happens at the motor end plate right here. So it's kind of weird because the motor end plate is this one spot on the skeletal muscle fiber. And what makes it the motor end plate is that like as opposed to the entire rest of the muscle fiber, this is the only spot that has acetylcholine-gated receptors. Acetylcholine is the neurotransmitter because this is a neuron. So acetylcholine is going to be released from this neuron across the gap and hit this neurotransmitter gated receptor that will cause a depolarizing effect that will stimulate an action potential, go both directions now away from this neuromuscular junction. That's what's going to be happening in this excitation contraction coupling step. And then when we release that calcium, we'll zoom in to the microscopic structure of contraction, the sarcomere, for our cross-bridge cycling. So first, let's think about what happens at the motor end plate. Do you know what I love about Brown Bear? That he's not brown, and that he is totally willing to be my eraser. Did you look at that? I couldn't find my eraser. So he said, "I'll be your eraser." Thank you, Brown Bear. You're usually the most mischievous in the house, but right now you're being the most helpful. Well, Baby Bear, Brown Bear just did all of the erasing. Next time, Baby Bear. Next time. Okay. No, I'm not erasing right there. Right now I'm drawing out numbers. So first, at the neuromuscular junction, this is where the lower motor neuron makes functional contact with the muscle fiber. So it's not actual contact because we have a synaptic cleft. But here's my muscle fiber, and it's full of myofibrils, okay, which are made of thick and thin filaments. And I'm not going to draw that out on this figure probably. The neuromuscular junction is where the neuron meets the muscle. There are 3 parts of a neuromuscular junction. 1, the lower motor neuron. 2, the synaptic cleft.
Mom.
Yeah, dear. And 3, the motor end plate.
Look at this in a mixer shop.
Look at it.
It's on your eraser.
It sure is. Where did I even find my eraser? The cement mixer truck has located the eraser, so Baby Bear and eraser can trade spots. Baby Bear wants to drive the cement mixer truck, and my eraser wants to come right back up here. Motor end plate. The motor end plate is this specialized region of the muscle fiber that contains ligand-gated receptors.
Oh, Kelly.
Oh, Kelly.
Hey, you broke your knee again.
It never got fixed. And the knee is the only thing that kept its original positioning. We're gonna pause. All right, the kids are at the plant store, so we're gonna go through this as quickly as possible. And I can only assume that I left off at number 3, the motor end plate. Because here we are, 1, lower motor neuron. So if we had to sum up what was happening in a lower motor neuron, we would already know this, where your other 201 counterparts wouldn't already know this, and they'd be like, I have to suspend my disbelief and call it a nerve impulse. What is a nerve impulse? An action potential, right? And what is an action potential? You have to answer this because not only do you need to know it right now for the lower motor neuron, but an action potential is about to be conducted down this muscle fiber. So if you don't know what an action potential is yet, you have to know it now. And you have to know it for the rest of your life as a knowledgeable person who knows something about anatomy and physiology, period. End of discussion. What's great is you can know it, because all it is that controls an action potential is a bunch of proteins that aren't thinking about anything. They're just doing what they do because they have no choice but to behave that way. Okay? So, what's an action potential? It is here coming down our lower motor neuron. If we are initiating a voluntary muscle contraction, where in the brain is that coming from? Oh, your primary motor cortex in your frontal lobes, right? In the precentral gyrus. That's why I do what I do, so that you know now I've got this upper motor neuron who's deciding to wiggle my finger. And if it's my right finger, then it's an upper motor neuron in my left primary motor cortex that's initiating that command. That upper motor neuron is going to talk to maybe an unnamed middleman. Either way, we're not naming that middleman, so we're not going to talk about it right now. What we're going to talk about is who we've always talked about and named, the lower motor neuron, who is synapsing onto the motor end plate. This lower motor neuron releases the neurotransmitter acetylcholine, which is acetylcholine, abbreviated ACH, which is how I'm going to write it out from now on. Acetylcholine, ACH, okay? So here, stored in my synaptic knobs, I've got all of this acetylcholine. And it's waiting for what? An action potential to travel down my lower motor neuron. So we're going to look at 3 parts of muscle fiber contraction and all of the steps involved there. Okay, so maybe I'll just write these first ones out in blue if you can see it. Let me make sure you can see it. I'm just going to stick with black, actually. So for everything that's happening at the neuromuscular junction, I'll write it in black, okay? So first, we have an action potential is going to travel down the lower motor neuron. What does that mean? Well, that I've got voltage-gated sodium channels opening and voltage-gated potassium channels opening and voltage-gated sodium channels opening, voltage-gated potassium channels opening. What does that mean? That I'm depolarizing, repolarizing, depolarizing, repolarizing, depolarizing, repolarizing. The entire way down the cell, this action potential is moving. And when I get down here to the synaptic knob or the axon terminal bouton, axon bouton, whatever you want to call it, this enlarged region storing my acetylcholine, the depolarizing swing of this action potential is going to cause the opening of these voltage-gated calcium channels. You already know this, right? It's a review. So the action potential travels down the lower motor neuron. The depolarizing swing opens our voltage-gated calcium channels, right? Right. You remember this stuff. So calcium's higher outside the cell than in. So I open my voltage-gated calcium channels. Calcium enters the cell, moves those regulatory proteins, and allows these vesicles to fuse with the plasma membrane, thereby releasing acetylcholine by exocytosis into the synaptic cleft. So, number 2, the action potential. The depolarizing swing opens our voltage-gated calcium channels. Calcium enters cell. Calcium entry causes exocytosis of neurotransmitter, specifically acetylcholine. I might not be doing this in the same number of steps as your book. It doesn't matter, and I don't want you to get mad if your book has 6 steps and your quiz has 6 steps and I only write up 5. It doesn't matter. I'm telling you that right now. You can understand it and you can break it down into however many numbers you want, okay? I don't know. You might get mad about that. I'd probably be mad about it sitting in my living room right now, but I'm not sitting in my living room. I'm standing in my living room teaching you about it. So, calcium entry causes exocytosis of acetylcholine. Acetylcholine is released to the synaptic cleft. Okay, so now I've got acetylcholine in here. And the motor end plate is this specific region of the muscle fiber that contains a specific type of receptor. It's the only part, or channel, it's the only part of the muscle fiber that has this specific type of channel. What might it be? If what I'm releasing is a neurotransmitter, which is a chemical also known as ligand, and that is acetylcholine. This channel might be an acetylcholine gated channel. In fact, it will be. So what we could say for our next step is that acetylcholine is going to bind a ligand-gated cation channel. What happens when a cation channel opens? This is a serious question, people. We have 2 cations move through it down their concentration gradients. Potassium is going to move out of the cell down its concentration gradient. Sodium is going to move into the cell down its concentration gradient. The influx of sodium is so much greater than the efflux of potassium that we will have a depolarizing event. We're going to call that here the end plate potential. In our neurons, it was an excitatory postsynaptic potential. You could still think about it the same way. It's excitatory, it's post the synapse, after the synapse, but specifically it's going to be an end plate potential because the synapse is ending at the motor end plate. Okay? So acetylcholine is going to be released into the synaptic cleft. It's going to cross the cleft and bind to acetylcholine gated channels here, and those channels are cation channels. So we'll say acetylcholine binds its receptor. And you're like, wait a minute, lady, you just told me that that was a cation channel, a ligand-gated cation channel. It was. The ligand is, and it is. The ligand is acetylcholine, and the receptor just happens to be a cation channel. So I'm going to come back up here and zoom in and recall that through a cation channel, sodium came into the cell, and it came in to a much greater degree or faster than the other cation that's moving through this cation channel, potassium, which is leaving the cell. So it's a cation channel. 2 cations are moving through it, but what's like physiologically really important is it's this net influx of sodium that's going to cause the end plate potential to develop. So that might be where, like, I think 6 in your book is like the end of this. 6 on my board right now would be like this opening causes the end plate potential to develop, but I guess I can just write that as the end of 5. So acetylcholine binds its receptor, which is a cation channel. Yeah, that makes sense. Yeah, perfect. The end plate potential develops. We're just going to pretend there was no doubt. End plate potential develops. Okay? Cool. What's the end plate potential? Potentials are changes in this like energy that we can measure if we stick a voltmeter in the plasma membrane. And so what happens as we get this net influx of sodium here is it's going to move us closer to threshold for the next part of our muscle fiber contraction, excitation, contraction, coupling. We have to get from excitation to contraction. So there are events that couple those things together, and that's going to be the conduction of an action potential, which is not what happened here at the motor end plate. In the motor end plate, we developed an end plate potential. Sure, it was as a result of an action potential coming down our lower motor neuron, but what happened right here was an end plate potential, okay? So I'm going to erase this board, and we'll, oh, nope, I'm not. I'm going to write number 6 that your book has, which is how we end signaling. So when you stop sending this action potential down this lower motor neuron, this acetylcholine that has bound its receptor right here and caused the end plate potential to develop is going to be either degraded or it will diffuse away. So it's degraded by an enzyme, so last step, 6. ACH is degraded by acetylcholinesterase, which is an enzyme, acetylcholinesterase. It takes it all apart and we're good. Acetylcholine is degraded by an enzyme and or diffuses away. And so if you diffuse away from the synapse, then you can't bind there anymore, and it doesn't matter because the only place that has acetylcholine receptors is the motor end plate. Okay? All right, so end plate potential. Let's just have a little bit more about that. The end plate potential, which I will from now on, it's probably the last time I'm going to write it, but from now on I'll abbreviate it as EPP. And this is the depolarizing event that occurs here when we open these acetylcholine-gated cation channels that will cause the action potential to be generated in our muscle fiber and the rest of our muscle fiber. So if we move away from the motor end plate, which is the only spot with acetylcholine gated channels, then in the entire rest of the muscle fiber I must have different channels. Those channels are the voltage gated channels that are now going to convert this chemical message here back into an electrical message that can be conducted into the muscle fiber. So that's the next step. So this action potential here causes this end plate potential here, which is going to generate an action potential in the muscle fiber that will result in our sarcomere shortening. Okay, how? Our sarcomere shorten when calcium is released. So we have to get from excitation to calcium release and cross-bridge cycling, which is happening here in these wonky, thick and thin filaments. It's fine, it'll work out. So the players that we have here, which since I drew the beautiful figure, I should have already like written out the key, but whatever, we'll just call it a good learning opportunity for you to review, are what? Let's just recall neurons. When our excitatory postsynaptic potentials, or depolarizing graded events, summed up to threshold, it was what threshold that we were trying to reach? Negative 55 millivolts, right? And that was the threshold of what? Our voltage-gated channels that were going to conduct our action potential. Our voltage-gated sodium and voltage-gated potassium channels. So, a couple things that we can just like put together here is that this end plate potential is going to be the net influx of sodium that occurs when we open this cation channel, acetylcholine-gated cation channel, and that net influx of sodium then will be this graded depolarizing event that will add up to open our voltage-gated sodium channels. So what we have here are our voltage-gated sodium channels in blue. And those, of course, are going to be responsible for the depolarizing swing of an action potential. These open at what? negative 55 millivolts. And they stay open until when? Positive 30 millivolts. Okay, and then what happens at positive 30 millivolts? Well, the other channels that I should have labeled before, but we'll just call it review, are what? What are the other important voltage-gated channels for an action potential? Voltage-gated potassium channels, right? They're going to give us our repolarizing swing. So our voltage-gated potassium channels are here in orange, and they are going to open at threshold, but we'll say they take over at positive 30 millivolts, and what are they going to do? They're going to repolarize the cell. How do we repolarize? Lady, you didn't even tell us the resting membrane potential for these cells. Well, hopefully you read about it, but if not, it's negative 90 millivolts, which is the equilibrium potential for potassium. So potassium wants to drag this potential to negative 90 millivolts. What happened when the voltage-gated sodium channel opened? How far did this potential go? Positive 30 millivolts. Potassium wants to get us from positive 30 millivolts inside the cell to negative 90 millivolts inside the cell. How does a positive ion do that? It leaves. I almost said it GTFOs, and then I did say it. It does! If a positive ion wants to take us from positive 30 in here to negative 90 in here, the only way that a positive ion can do that is to leave the cell. And it will, that's the repolarizing swing. So, at positive 30 millivolts, our voltage-gated potassium channels take over, and potassium exits to negative 90 millivolts, which is its equilibrium potential, which is also resting membrane potential. That's really, I mean, as important as it is, horribly important. Horribly important. It's not like really that interesting. I mean, it is, but like as far as signal conduction and stuff is not really that interesting, because what's happening now is this depolarizing swing that's going, we're depolarizing this one because we reached negative 55. This one's going to positive 30. Well, that's way over negative 55. So this one's going to positive 30, going to positive 30, going to positive 30. That depolarizing swing is really the interesting part, because that's the business that gets stuff done right now in this cell. And the stuff that we need to get done right now in this cell is we need to get some cross-bridge cycling done. We need to get these myosin filaments to grab those actin filaments, do some sliding, we need to shorten, we need to generate tension, we need to contract this muscle fiber. So how we're going to do that is we're going to let all of this calcium that's stored here in what? The sarcoplasmic reticulum, right? This modified smooth endoplasmic reticulum of our skeletal muscle fibers is storing all of this calcium. And it's this depolarizing swing that's going to open these voltage-gated calcium channels and allow calcium to exit the sarcoplasm and exit the sarcoplasmic reticulum and flood the sarcoplasm, so that now we can remove the inhibitory action we have on our thin filaments and we can start that cross-bridge cycle. It is so exciting, and it's one of my favorite things that happens. So don't be intimidated. Rewind and slow down to 50% if you need to. I hope you're going to love it. I know that you love contracting your muscles. Whether you know that you love it or not, you love it. And so let's get down there. We generated our action potential by first developing an end plate potential here. If we needed to write some words, we could say that this net influx of sodium through the cation channel is going to be a depolarizing event that opens our, voltage-gated channels because we reach threshold. So we could say the end plate potential causes the cell to reach threshold. And then the next thing that we're going to happen is excitation contraction coupling. And what we could say occurs in excitation contraction coupling, I think I wrote the last one in black. I'll write this one in green. I know that it wasn't green. So for excitation contraction coupling, we could say that number one, the end plate potential stimulates the opening of our voltage gated channels in the sarcolemma. Okay, number one, great. The action potential is that it's going to stimulate the opening of our voltage-gated channels. Which ones? Voltage-gated sodium channels, depolarize. Voltage-gated potassium channels, repolarize. That is an action potential. So now our action potential is going to be conducted down the length of the sarcolemma and the T-tubules. Action potential is conducted down the sarcolemma and T-tubules. Okay, wunderbar. The action potential, the depolarizing swing of the action potential is going to open our voltage-gated calcium channels in the sarcoplasmic reticulum. So depolarization opens voltage-gated calcium channels. In the sarcolemma and T-tubules. Now, when we got down these T-tubules, this was part of what? A triad, right? We said that one T-tubule and two terminal cisterns of sarcoplasmic reticulum formed a triad. So this action potential is going to be conducted down the sarcolemma and down the T-tubules. And when it comes down those T-tubules, it's going to stimulate the opening of our voltage-gated calcium channels. Okay, so I will write that next step up here. I'm realizing you can't see it right there. So this last step, depolarization, opens our voltage-gated calcium channels in the sarcoplasmic reticulum. So this depolarization opens our voltage-gated calcium channels in the sarcoplasmic reticulum. Calcium is released to the sarcoplasm, and now we're going to be able to begin contraction. So we have coupled excitation to contraction by generating and conducting an action potential. So for this last step, we'll say depolarization is going to open our voltage-gated calcium channels in our sarcoplasmic reticulum, and calcium will flood the sarcoplasm. I am 100% sure your book does not have that broken down into 3 steps. It's probably like 4 right here. And a moment ago I had all 4 steps, but my children just returned home and I see my car pulling up and there's a storm coming and all the lights changing and I'm just like, we have to get through this lecture. It's take freaking a million, people. So here, cut. And we're gonna get through it together. Somehow I feel like I'm missing a slide I need, and again we're going to race through this. So just quickly to make sure that I know where we're going. What we have done is at the motor end plate we had an action potential traveling down our lower motor neuron, causing the release of acetylcholine into the synaptic cleft. It crossed the cleft and bound to acetylcholine receptors on the motor end plate. At the motor end plate, we developed the Endplate Potential, which occurred as the result of a net influx of sodium through the cation channel. That is going to allow us to reach threshold for our voltage-gated channels in our sarcolemma and T-tubules. So when we reach threshold, voltage-gated sodium channels open. Sodium dumps into the cell, depolarizing it. At positive 30 millivolts, that sodium channel closes. The potassium channel takes over and repolarizes the cell. We conduct down away from the motor end plate in both directions. So this is important. And the resting membrane potential, there's not an absolute refractory period in skeletal muscle like there was in neurons. The resting membrane potential of our muscle fibers is the equilibrium potential of potassium. Okay, so in both directions away from the motor end plate, we send this action potential, which means that in both directions away from the motor end plate, we can start this action potential conducting down that sarcolemma and T-tubule so that calcium can get out, and in both directions away we can start contracting or start cross-bridge cycling, which is going to allow for shortening and contraction of our skeletal muscle. And it has to happen in both directions away because we want the entire thing to shorten at once. And so I feel like I'm somehow missing a slide again. So, because we've already talked about generation of an action potential, and we left off saying that the depolarizing swing of the action potential down the sarcolemma and T tubules was going to cause the opening of voltage-gated calcium channels in the sarcoplasmic reticulum. So that was excitation contraction coupling, or ECC. Excitation contraction coupling. And that is how we go from an action potential, an action potential at the neuromuscular junction, to the second step, excitation contraction coupling, ECC, the conduction of the end plate potential to an action potential in the skeletal muscle fiber, to actual muscle contraction. Okay? That's our third step. What we're trying to get to is muscle fiber contraction, how one individual muscle fiber shortens. And what we have to do is get calcium out. And so what happens then, where we left off, we said calcium floods the sarcoplasm. Now zoom back in your memory to when we were talking about myofibrils and myofilaments, and we said that calcium was going to bind to what to do what? Like, man, lady, I don't even know what you're talking about. Calcium is going to bind to troponin to move the troponin-tropomyosin complex. So calcium is going to flood the sarcoplasm. Calcium binds troponin. What this is going to do is move the tropomyosin complex. And what that does is it exposes the myosin head binding sites on actin and leads us to the third part of muscle contraction, the muscle fiber contraction. 3, the third big important step, because all of this started, even though I'm missing a slide, with muscle fiber contraction. 1, what happened at the motor end plate was what happened at the neuromuscular junction. 2, what happened down the sarcolemma and T tubules, or the generation of an action potential. And now 3 doesn't have its own slide, but the other third most important event in muscle fiber contraction is the cross-bridge cycle. And what is going to occur in the cross-bridge cycle is that we're going to form these things called cross bridges, where myosin reaches up, forms a cross bridge with actin, grabs onto the myosin head binding site on actin, forms a cross bridge. It's going to pivot, pull our thin filaments toward the center of the sarcomere, then it's going to release, cock back, do it again, over and over and over. Cycle as long as calcium and ATP are present. So 3 important steps involved in contracting a muscle fiber. The steps and events that occur at the neuromuscular junction, which is excitation. The steps and events that occur at the sarcolemma and T tubules, which is excitation, contraction, coupling. And the steps and events that occur at the sarcomere, which is the cross-bridge cycle. Okay, so this is what's happening at our sarcomere. So we've got our Z discs with our thin filaments right here, and we're going to cycle through our thick filaments, pulling them toward the center. So that if this is what we look like when we're relaxed, that what we're going to look like at the end of it is that we have all this kind of overlap, because we have shortened our sarcomere, and we've shortened every sarcomere in a muscle fiber, shortened every muscle fiber in a fascicle, shortened every fascicle in a muscle, and you can do a whole lot of different stuff. Okay, but before we get to that, let's look at the mechanisms of this. So really what happens in the cross-bridge cycle is a cycling of 4 steps, and they can occur when calcium binds to troponin and moves the tropomyosin complex. Now what happens is our myosin head binding site on actin is exposed. Myosin head binding site exposed. So the first step in the cross-bridge cycle And that's why I kind of went back and summed it up with all numbers again, because you've got first, second, third steps in every one of the first, second, and third steps. Sorry people, it's what we gotta do. The first step in the cross bridge cycle is how we get the name. It's cross bridge formation. And what's gonna happen is myosin is gonna reach up and grab onto the myosin head binding sites on actin, forming a cross bridge. Number 1, cross bridge formation. And if we wanted to write words about it, we would say myosin grabs myosin head binding site, forming a cross bridge. So if we were to zoom in to like one individual thick filament and what's going on here, they're sitting in this like high energy conformation. And so what happens now is they're going to reach up and grab on. Okay? Cross bridge formation. I reach up, I grab on. That's my cross bridge. The second step is called the power stroke. I'm actually going to erase all the rest of this. The weird thing about calling it a power stroke is that it makes it sound like it requires some power, and it does not. ATP is not required for the power stroke. Myosin, when it is just sitting here relaxed, is already in its high energy conformation. So all of the energy that it needed to get there has already been done. This is where my shadow is working well and I can see it. So myosin is sitting here in its high energy conformation. When we expose our myosin head binding sites on actin, it reaches up and forms a cross bridge. Okay, in the power stroke, it just goes to its low energy conformation. This is where it wants to be. So this is the second step in the cross bridge cycle, is we could say, they call it the power stroke. And it's just a misnomer of a name. And they'll get you on that on the MCAT. So if you're a pre-med student, beware of that. The power stroke does not require ATP. So what happens in the power stroke is that myosin is going to pivot. If what it looks like in its high energy conformation is like that, it doesn't want to be there. Nothing wants to be in the high energy conformation. Everything wants to be low energy. So what it does when it pivots is it just comes back to where it wants to be. It goes from there to there. It goes from there to there. And when it goes from there, if it reached up and grabbed actin and it goes to there, then it pulls that actin, okay, from there to there. And I move my Z-disc a little bit closer to the center of the sarcomere on that side. And I move my Z-disc a little bit closer to the center of the sarcomere on that side. So in my power stroke, what I could say is my myosin pivots to the low energy conformation. And what happens is, how we would finish that sentence appropriately, is pulling actin, or pulling the thin filaments toward the center of the sarcomere. Okay, now this is the part that requires ATP. And this is the part that is kind of hard for people to wrap their heads around sometimes. The third step of the cross-bridge cycle, after we have pivoted, is to release, and this is where we need ATP. So to let go of your contracted state, you need ATP. You need energy to let go. That could probably be a metaphor for life. So as you let go, then we bind ATP, let go, so we release, that's the third step, and then we can hydrolyze that ATP we just bound to reset to the high energy conformation, the fourth step. So let's write out number 3 is that ATP binds and myosin releases. So if we needed to name it, because we said 1 was cross-bridge formation, 2 is the power stroke, 3 is the release. Okay, and that is going to happen when ATP binds and myosin lets go of actin. And then in our fourth step, This is reset. Or a lot of books will say it cocks back. So what happens if I'm myosin and I have bound ATP, I can burn the energy in that ATP to reset to my high energy conformation. So for reset, I use ATP to cock back. Hold on, folks. Cock back to high energy conformation. And now I'm here. And if calcium is still present, then troponin and tropomyosin are still out of the way, and I'll reach up. And number one, I'll form another cross bridge. And then number two, I will move to my lower energy conformation and pivot, or do my power stroke and pull my Z disc toward the center of the sarcomere. And then if ATP is present, I'll bind it and I'll release, and number three, I'll release, and then number four, I'll burn it and I'll reset, and then number one, I'll reach up in cross bridge formation, and then number two, I'll power stroke, and then number three, I'll release, and then number four, I'll reset over and over and over again in a cycle as long as calcium and ATP are present. Okay, so the cross bridge cycle, the big important takeaway we could say here is this is going to continue as long as calcium, particularly, is present. And then in parentheses, we could say ATP. Okay, now here, so wait a minute. Let's think about this for a minute. ATP is what's going to cause release or relaxation. So when you die, rigor mortis occurs because you cannot relax your muscles. The moment that you die, every cross bridge that is formed and have performed a power stroke is stuck there in that power stroke, holding that muscle in whatever position it was, because you can't bring any more ATP there to let go and then cock back. Okay, so that's what causes rigor mortis. Pretty interesting. So then you say, well, how then can you move like cadavers and stuff? Well, because then after a couple days, decay takes place and we do let go, but it's not because of a natural cycling of events. Okay, so that's just something to be aware of. And then again, remember, well, as we discussed last time, the analogy with the zombie, right? That it's not like every single one is grabbing and letting go at the same time. So they are all in different phases of the cross bridge cycle, so that while one is grabbing, one is releasing, and the next is resetting. And they're all working together to bring these sarcomeres, or to bring these Z discs together, shorten our sarcomeres, and generate tension in our muscle. Okay? Okay. The following tissue is subject to conscious control. A, skeletal muscle. B, cardiac muscle. C, smooth muscle. D, all of the above. Yeah, you're right. It's a skeletal muscle. Okay, in order to receive a signal to contract, each skeletal muscle must be served by a or an artery, a vein, a nerve, or all of the above. Yeah, the answer is a nerve. And you're like, wait a minute, lady, you need all of those to contract. So I would say, yes, you do need all of these to contract, but the question is not asking you, what does a skeletal muscle need to contract? It needs you not to have your microphone cord wrapped around your ankle. It needs all of the above to contract, sure, because you can't contract if you're not living, and you can't live without an artery and a vein. However, the question is saying to receive a signal to contract. Guess who's not sending signals? Arteries and veins. Nerves send those. And so, yeah, that's it. Ba-dang dang. All right, the following proteins are found in thin filaments. A, actin. B, tropomyosin. C, troponin. D, all of the above. Yeah, you're right, all of the above. Whoa. All right, the motor unit. How is it possible that I can do things like scratch my forehead with this one finger, or hold this marker with this one finger, or hold this heavier bottle full of fluid with this one finger. How much could I make this one finger do with my living room right now? Could I hold this entire lower limb with this one finger? Ow, if it wasn't pinching me, I could. Yeah, I can. How can I make this one, how much can I make this one finger do? Okay, well, I can recruit, like, man, lady, there are so many different muscles involved in the holding of that bone on your finger. It's true. We're going to suspend our disbelief and believe it's only this one finger, and that in order to get from here to here, what we were doing was recruiting more motor units. So, ow. You know that you can do a bunch of different stuff, and some of it requires a lot from you, and some of it requires next to nothing. And so how we do varying levels of work in each muscle is by recruiting varying motor units. So the definition of a motor unit is that it is one lower motor neuron, or the axon terminal of the, it's one lower motor neuron and all of the muscle fibers it innervates. So let's write that down and talk about it. One lower motor neuron and all of the muscle fibers it innervates. Okay, well, in your book, you've got a figure that is showing you two different motor units. And so we've got, let's say, our pink lower motor neuron. And it has all these telodendria, right? These axon, there's like branching of its axon that's going to give it multiple axon terminals. Okay, so this first pink motor unit has our pink lower motor neuron and these two pink muscle fibers that it is innervating. Okay, so when the signal comes down from my upper motor neuron to this lower motor neuron, both of these muscle fibers are going to contract. Okay? All right, well, I also could have another motor unit here, my blue motor unit, and it's got 1, 2, 3 muscle fibers in it. And so what happens is that if you zoom into its end, its telodendria are branching, and we have these 1, 2, 3 terminals. So when the command coming down from my upper motor neuron stimulates an action potential in this lower motor neuron, it's going to stimulate the contraction of 1, 2, 3 muscle fibers in that motor unit. Okay? Now I would never see some motor unit where I've got, let's say, a pink, I would never see it happen where a pink neuron terminated on a blue muscle fiber. That just couldn't happen, because then you would get confusion in all of your signaling and you wouldn't have efficient contraction. So this is important, and it's weird because it's a little nitpicky. You can have one lower motor neuron that innervates multiple muscle fibers, but you cannot have one muscle fiber that is innervated by more than one motor neuron. Okay? Because my muscle fibers don't branch in the same way that my neurons do. Well, I mean, that's like an explanation. That's not why, but I mean, that's just like physiological or an anatomical, a numbers reason. But also because it wouldn't make sense. I wouldn't want this blue muscle fiber to then like maybe say also be responding to some green motor neuron. Because then how would this muscle know any difference between green motor neuron firing and blue motor neuron firing? It has to know the difference so that I can do different things, and I can recruit more motor units as I need to do various tasks. Okay? I hope that made sense. Look it over in your book. Ask me questions in the discussion page if you need to. Here we go. Okay, we said that we were using a model, a sliding filament model of contraction to try and understand contraction because it's so small that we can't actually observe it. Well, if we can't, well, in a single muscle fiber. If we cannot observe it in a single muscle fiber, what can we observe? The behavior of an entire muscle. So I can take a frog gastrocnemius muscle, which is where? Your calf, right? And I could hook it up to a tension meter and I could stimulate it with varying levels of stimulation at either varying frequencies or varying level of stimulus, and I could watch the effect of contracting that entire muscle. So I could see the whole muscle shorten, and that would be a muscle twitch. I couldn't, like in a living muscle, I can't watch one individual muscle fiber contract. So in order to take all of these facts that I know and put them together in something I can understand, we look at the behavior of a muscle. So let's just imagine right now that I'm just like, I'm gonna send down one command to my biceps brachii, and then it's just gonna be long enough to contract and relax. Then what's gonna happen is my muscle is gonna twitch, right? I wonder if you can see it. I can see it. Okay, so I twitch, I generate tension, I relax. Okay, so what happens in a muscle twitch, if you look, let's look at a graph of this and say what we're looking at time here and tension here. Okay, and the point of stimulus, or the point at which I send down this command, this action potential down my lower motor neuron, is right here. Okay? Then what I first observe before I see any tension is that I actually see a latent period. And this is the time between stimulation and the development of tension. So latent period is the time between stimulation and the development of tension in the muscle. Now, what might be happening physiologically? There are three important events in the contraction of a muscle fiber. What we're doing now is contracting a bunch of muscle fibers in a muscle. If we're saying that what's happening at the micro level is happening at the macro level, then what event might be occurring while I see no tension? Excitation, contraction, coupling, right? My action potential is traveling down my sarcolemma and T-tubules. My calcium is entering my sarcoplasm. And now as it starts binding to my troponin, and I'm moving the inhibitory regulation of those proteins, then I will start to see the development of tension. Before that happens, I see no tension. This is the latent period. So we could say this is the time between stimulation and the development of tension in a muscle. And physiologically, the events that are happening are excitation, contraction, coupling. Why do I know that those are the events that are happening right here? Well, because right here at the start, that's excitation. Where's that occurring? The neuromuscular junction. Okay? Right here is my excitation, contraction, coupling. And that's occurring down where? My sarcolemma and T-tubules. And then, when I start to see the development of tension, what is physiologically occurring in my muscle fibers? The cross-bridge cycle, right? Right. So, as calcium enters the sarcoplasm during excitation, contraction, coupling, and starts binding to the troponin and moving the inhibitory interaction of the tropomyosin complex, then myosin can start reaching up, grabbing onto actin, pulling my sarcomeres together, and developing tension. Okay? Now in a muscle twitch, if all I do is give you one stimulus, and then relax, just one brief moment of stimulation. For a muscle twitch, I'll see that I generate tension, and then I drop off, I have a relaxation phase, and I come back to rest. Okay, so the latent period occurs when I am doing excitation, contraction, coupling. Then I have my period of contraction, and what we could say for my period of contraction is that this is like this time, well, kind of all this time, this is the time during which the tension is developing in the muscle. Tension develops in the muscle. And then I get my period of relaxation, and that occurs here after, because all I'm doing is twitching, right? One single stimulus. So it's enough to allow some calcium, I generate some tension, and then I'm gonna reuptake that calcium and my tension is gonna drop off. I'm gonna relax. So during this period of relaxation, we could say what's occurring here is that calcium is being actively transported back into the sarcoplasmic reticulum. Calcium actively transported back to the SR. Okay? All right, that's a muscle twitch. So I have an action potential coming down my lower motor neuron. It stimulates the opening of my acetylcholine-gated cation channels. The net influx of sodium generates an action potential that's conducted down my sarcolemma and T-tubules that is going to result in the opening of voltage-gated calcium channels in my sarcoplasmic reticulum. Calcium is going to enter the sarcoplasm, bind to the troponin on the troponin-tropomyosin complex, removing the inhibitory interaction so that myosin can reach up and form a cross bridge. We'll do the cross bridge cycle, we'll generate tension, and then as soon as I'm done sending down my action potential, there's no more stimulus, I'll actively pump that calcium back into the SR, and I will relax, and that is a muscle twitch. Okay, now what about when I am doing stuff? Because it's not very often you just sit around and you're like twitching. Like, come on, what's that physiologically appropriate for? Almost nothing other than demonstrating muscle twitch in an A&P class from your living room. So instead, what is relevant is how we can grade muscle responses. And again, it's not like, in a moment-to-moment basis, everything is kind of a mixing of everything that you can do. But in a scientific classroom where we're trying to understand how things work, we can break it down into different steps. And so if we look at graded muscle responses, we can either change how often we stimulate the muscle, or we can change how strong we stimulate the muscle. And so if we're changing the frequency of stimulation, we call this wave summation. And what happens in wave summation is that you're going to build on your tension in a wave-like fashion. So wave summation occurs when tension builds in a wave-like fashion as you increase the frequency of stimulation. And the reason it's occurring is because you're decreasing the period of relaxation so that not all of your calcium is being returned to the SR. Maybe it'll make sense when we look at a figure in a minute. So wave summation. We could say this is adding up in a step-like fashion due to increasing the frequency of stimulation. So we add tension in a step-like or wave-like fashion. And this is due to increasing the frequency of stimulation. And what that does is decreases the relaxation period, or the period of relaxation in our muscles. Okay, cool. So what different ways can we sum our waves? Well, actually first, we can get like this stepwise. So let's say here, okay, here's our graph. And here is our muscle twitch. We stimulate there, we have a latent period, we generate contraction, we relax, okay? Well, now we're gonna do wave summation, and this first thing, we're gonna call it trapé. So I'm gonna stimulate here, but I'm also gonna stimulate here, and then I'm also gonna stimulate here. So what happens is that I stimulate here, I have my latent period, okay, then I generate tension, then I start to, generate tension, I start to relax, and then I stimulate again here. And so, then I'm gonna generate tension, but then I'm gonna start to relax. but then I stimulate right there. So then this develops what we call trepe, which means stairs or steps. And this is a stepwise or a step-like development in tension as we increase frequency of stimulation, but allow for a small period of relaxation. So trepe is a stepwise increase in tension as we increase the frequency of stimulation, but still allow for some relaxation. So we're going to still allow some, we'll say we still allow a brief period of relaxation. So we'll start to relax, but then we're going to stimulate it again. So because we didn't fully transport all of our calcium back into the SR, there's still some tension in that muscle. And now we're going to stimulate it again. So we're going to release more calcium. We're going to develop more tension. And then we'll start to relax, but then we're stimulating again. So we haven't fully pumped back all this calcium. We haven't fully pumped back all that calcium. So if we stimulate again, we'll release more calcium. We'll develop more tension. And so trepe is a stepwise increase in tension as we increase the frequency of stimulation. But we're still allowing for some relaxation. Okay, the next thing that we'll see is that we'll reach what's called unfused tetanus. Ultimately, what we're trying to get to is a tetanic contraction, where we can fuse our tension and maintain some tension to do some specific activity. But what you see in unfused tetanus is you will increase your frequency of stimulation so much that you'll develop tension, but you won't quite fuse it into a wave. So all I want you to know for unfused tetanus is that we are further increasing the stimulation above trepe. So we'll increase the frequency of stimulation above trepe, but still allow for a brief moment of relaxation. And of course, my card ran out of space. Now I got a crying child and it's all falling apart. So I think that the only spot I lost was fused tetanus. So this is what we're trying to get to when we're trying to do something like maintain a long-term kind of contraction or action. And I really think I did unfused tetanus in the video that wasn't lost. I was all the way through this, people, and I was going to get to put my child to bed on his own. Instead, it says he has to really traumatize. What happens in fused tetanus is we are fusing tension. Okay, so that we have a constant amount of tension so that we can do, a lot of activity. I don't know. The example I had given earlier was I walked around like a fool holding this thing up. And I even talked about how, well, like when one motor unit gets tired, you gotta like activate another motor unit and blah, 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 blah. The point is, in fused tetanus, what happens is you fuse your tension. So again, we're looking at time and tension, and if here's our point of stimulation, then our first point of stimulation, then we'll see a slow latent period. But now what we're doing is there is absolutely no period of relaxation. So each consecutive stimulus is coming so quick that after my latent period, as soon as I start developing tension, I'm just going to keep developing tension. And this will occur until I stop stimulating the muscle and can actively pump all that calcium back in. So then during this period of relaxation, I'd be actively pumping all of my calcium back in, or your muscle reaches fatigue. If you reach fatigue, you'll also have a drop off in tension. I can't really say more about it right now because I'm so frazzled and tired and tetanic, I'm sure. So you can read about it in your book. I think that my explanation was probably good enough. Fused tetanus. a constant amount of tension that's delivered because we increase the frequency of stimulation so high that there is no period of relaxation. So we increase the frequency of stimulus so there is no period of relaxation. And what that will do is allow us to generate constant tension. Either until I stop sending the command, stop the stimulus, and then allow myself to naturally relax, or until I fatigue. I am fatigued right now, and I lost my pointer in my very hand. So the other thing that we can do is, instead of changing the frequency of stimulus, I can give you one stimulus and allow you to generate tension and relax, but I can change how strong that stimulus is. So this is like the difference between like catching like this faint smell of a lilac as you walk by somebody's yard versus like some 50 pumps of cheap dollar store perfume all over your grandma's face when she comes to kiss you with her unwaxed mustache. I don't know. So that like subthreshold you wouldn't detect. At threshold, that's the small lilac you're walking by. Lilac, lovely. Like maximal, you're like, oh grandma, get away with your crappy cheap perfume, please. I love ya, but from a distance with that stench. Okay. These are relevant examples, people. Just always tell yourself that. I have paid for these relevant examples. So the other way that I can grade a muscle response is that I can change the strength of stimulus to that muscle. And what occurs then is what I call motor unit recruitment, well, what everyone calls motor unit recruitment. And as you require a muscle or whatever, I mean, so your nose wasn't the muscle, but you know, like it's relevant and relates. My guy's crying, it's breaking my heart. So as you need to do more and more, you're going to recruit more and more motor units. And if, let's say what I'm doing is that I've taken out a biceps brachii and I hook it up to this tension meter. And what now I'm doing is I'm going to have two graphs. On the top graph I have tension. Honey, you don't have to go to bed until 7 o'clock. You can play with all your toys.
But he wants to play with you.
Well, I am teaching a class, and I love you with all my heart. And on the other graph, I have level of stimulus. Sorry. So, level of stimulus. Okay. And at sub-threshold levels, let's say this is threshold. At sub-threshold levels of stimulation, so I stimulate my muscle, here we are at rest, and I give it like 1 millivolt level, or really, really low levels of stimulus. I generate no tension. Why? Because I'm below the threshold. So this is any level of stimulus, which is just a change in a variable. So I could have a change in level of odorant that I'm not aware of, but a dog might be, right? So subthreshold stimulus, this is a change that is below our detectable level. Or so in a muscle, like if there's a feather on my hand, I don't have to recruit any motor units in my muscle to hold that feather. Why? Because my bones are strong enough, darn it, they're holding up the whole level of tension of the muscle, right? Okay, so a feather, like if what we're looking at is not something like smell, which is nervous system, instead something like muscle, which is what we're talking about, then a sub-threshold level of stimulus would be placing like a feather on this finger. I don't have to require any muscles, or on this hand. I don't have to recruit any motor units in any muscle to hold that feather, because just the very nature of me is holding all of me up right now. So it's not like I'm doing anything extra. So the tension will be that there's no tension. Okay, well now, if it's just this one finger and now, like, okay, I put an eraser on there. Sure, now there's some more weight to that. and furthermore, I've got to balance it. So now, like, all of these various motor units are going to come on and start responding. So what we'll see then is our threshold stimulus is the first level of stimulation that is going to result in my muscle generating tension. And so what I'm doing each time is I'm only giving you one single brief period of stimulation, so it's enough to cause a muscle twitch or not. So I'm allowing a period of, in theory it would be down here like a period of tension, but there's none in the relaxation. Here I'm just at my tension, so I'll cause a twitch, I'll generate my tension, my threshold level of tension, and then I relax. Okay? Okay. So threshold stimulus is the first or the level of stimulation that recruits the first motor unit in the muscle. What's a motor unit? One lower motor neuron and all of the muscle fibers it innervates. So let's imagine that this is my biceps brachii and this is a cross section of it. So I've cut it in half and I'm looking down at it. Okay? So in it are all of my fascicles of muscle fibers that contain motor units, right? Okay, at subthreshold stimulus, I am not going to recruit any motor unit in this muscle. So if I ask my biceps brachii to hold a muscle, or to hold a muscle, it'd be like I am a muscle lady. If I ask my biceps brachii to hold a feather, it'd be like, I don't even have to think about that. I don't have to recruit any motor units to hold that. Like that level of stimulation is so low that it requires no tension from me. Okay, well, what if I want you to hold a can of tomato soup? All right, well, that's the threshold that I'm actually going to require my first motor unit to be activated. So this is subthreshold, and I've recruited no motor units. Now, at my threshold, I recruit the first motor unit, and if it's my biceps brachii, let's say what takes the first motor unit is a can of tomato soup. Is this my most or my least sensitive motor unit? It's my most sensitive, right? So the least amount that I'm ever asking this muscle to do is going to recruit this first most sensitive motor unit. Okay? So that's threshold stimulus. This is the level of stimulation that recruits the first motor unit in the muscle. It's the most sensitive. Okay, great. Change colors, lady. I'll do it. Now, if I deliver it instead of a can of tomato soup, I deliver it a 10-pound dumbbell. Then what's going to happen? Well, so here is my level of stimulus. Then I'm going to relax. And now I'm going to pick up a 10-pound dumbbell. Somebody's going to give me one right to my biceps brachii. So now I'm going to develop more tension because I'm requiring my muscle to do more. But I'm still only giving it a 10-pound dumbbell for 10 pounds of stimulation, you know, that level, higher level of stimulation for brief enough to have a twitch, and then I relax, okay? And I come back. So now I'm above that, and now I had already recruited my most sensitive motor unit, and now to carry that dumbbell I'm recruiting another motor unit, and so now this muscle is generating more tension, and I don't have enough space to go real far on my board. So let's just say the next thing that we do is we give my biceps 45 pounds of stimulation at once. Okay, so that's its max. That's the most my muscle can do. And so let's say that's the level of stimulus is all the way up here, and I give it 45 pounds. Then I develop all of the tension that my muscle can develop. I recruit my most sensitive motor unit, and I recruit the next motor unit, And I'd recruit the one that wasn't labeled in here, and the next that wasn't labeled in here. And then when I hit it with its max stimulus, I'd recruit every single motor unit in my biceps brachii to do this job. So when I recruit that last one, is it the most sensitive or the least sensitive? It's the least sensitive, right? So now I have recruited every motor unit in this muscle, and it's producing the maximum amount of tension that it can. Okay, so I'm some poor frog hooked up to some scientist's test thing to, you know, teach some students some lesson, and the next thing that they do is they deliver 50 pounds of stimulation. Can I develop any more tension? With a higher level of stimulation beyond the recruitment of my last least sensitive, the maximal level of stimulus my muscle can respond to, can I generate any more tension? The answer is no. So my maximal level of stimulus is the level of stimulation that's going to recruit every motor unit. So now if that's 45 pounds for my biceps brachii, and I hit it with 50 pounds, I can't generate any more tension here. And I mean, and this doesn't quite work in humans, but if I'm a frog gastrocnemius or a frog biceps brachii, then I can hit with 50 pounds here, and I'll still only generate this much tension. 55 pounds here, still only generate this much tension. Why? Because as soon as I activate this least sensitive motor unit, the last motor unit of this muscle, it cannot do any more than what it is doing for you now. So the maximal stimulus is the level of stimulation that recruits every motor unit in the muscle, and we produce the most tension the muscle can do. So the level of stimulation that recruits every motor unit in the muscle. So this will produce our maximum tension. Okay, people, we're really racing through this last part because I got a broken heart over there that I want to put to bed. So, isotonic contractions. Iso, same, tone, okay. So isotonic contractions, what's happening is we are in an isotonic contraction, we are changing the length. In an isometric contraction, iso, same length, we're not changing the length. So in an isotonic contraction we are, and there are two types of isotonic contractions, but first let's say in an isotonic contraction, we change the length of the fiber or the muscle when generating tension. And we can do this two ways. We can shorten the muscle when generating tension, which is something that what we most often think of. Like when I flex my forearm, I'm shortening my biceps brachii in a concentric isotonic contraction to do that. Now, there are also times when I lengthen the muscle, and we'll talk about that in a minute. So in a concentric contraction, what I'm doing is the change in length is that I am shortening. So I shorten the muscle or the fiber, and generate tension. Okay, and that's the one most people understand. But it's also possible that I do an eccentric contraction, and in an eccentric contraction, I'm actually lengthening the muscle or the muscle fibers of the muscle as I'm generating tension. So let me write that out and then we'll talk about it. So I lengthen the muscle or the fiber and generate tension. So in both of these, I'm changing the length and I'm generating tension. but I'm doing it in opposite ways. I think the easiest, like most like practically relevant way to understand an eccentric contraction is, imagine you're walking down a hill, like a really steep hill. You're performing a bunch of eccentric contractions in the muscles in your thigh to do that. Hang on a minute. So a really great example of when you're doing concentric and eccentric contractions together is when you're doing like biceps curls, or if you've got like a thorax or a baby and you're like lifting them to your face to smooch their face. Then when you lay them back down, like there's also some tension that's required to maintain their body weight. So when I'm lifting the baby up to my face, the thorax, you speak for the trees, you can have your own kisses. If I'm lifting the thorax to my face, my biceps brachii are doing a concentric contraction and they're shortening to generate the tension to do this. And now, when I am laying the thorax back down, my biceps brachii are performing an eccentric contraction and still generating, there's still tension. Okay, so if we look on the opposite side of that, when I'm in a concentric biceps contraction, my triceps are doing an eccentric contraction, and then they're doing a concentric contraction while my biceps are doing an eccentric contraction, and those are my isotonic contractions. Thank you, Lorax. There's little man, and we've got like 2 more slides. Okay, isometric. Iso, same, metric, length. Isometric contractions are those contractions in which you're generating tension, but you are not changing the length of the muscle. And here to help us illustrate is brown bear. So in an isometric contraction, like if I wanted right now, you could kind of think of it, hold on, let's step back. In an isotonic contraction, the work you're asking the muscle to do is not greater than the work that it can do. So it changes length and it moves that load. In an isometric contraction, the work that I'm asking the muscle to do is greater than it can do. So the muscle can generate all sorts of tension, but I'm not changing the length of those fibers and I'm not moving the load. So brown bear can push on this wall as much as his little big attitude can make him do. And he's generating tension, sure, there's a lot of tension, but his muscles aren't changing length to generate that tension. So what we could say in the isometric contraction is that the work exceeds, or the work or the load, the load exceeds what the muscle can do. So you'll generate tension, but you're not going to, you have no change in length. And here to help us finish out with these last 2 questions, or these last 2 slides, is elephant in my armpit. Okay, so hello, elephant. Welcome to class. All right. A muscle contraction increases in strength to a certain point because A, recruitment occurs and more motor units are activated. B, additional neurons begin stimulating each muscle fiber. C, more calcium is available in the sarcoplasm. D, all of the above. Or E, A and B only. This one's kind of tricky. It's not tricky, it's thought-provoking. None of them are tricks. It's just to get you to think, people. The answer is A, recruitment occurs and more motor units are activated. And so if we look at these answer choices, you could say, well, why isn't it this? Because that's the one a lot of people pick. And then some people actually, a lot of people also pick all of the above because they're like, well, more calcium is available in the sarcoplasm. All right. No. Okay. The physiological mechanism that's allowing our muscle contraction to increase in strength is that we are slowly getting one muscle fiber after another muscle fiber recruited. And we do that as we bring on more motor units. Okay? So that's the answer. Additional neurons will never begin stimulating each muscle fiber. Remember, muscle fibers are only innervated by one lower motor neuron. But one lower motor neuron might innervate more than one muscle fiber. All right, it's bedtime. Good night, people. This channel might not, I mean it might, but it's not, it might not. This. All right, my people, we're really getting through this lecture this time. And yes, there may be thunder outside, but I'm confident I can be louder than the thunder, and it doesn't matter about all the shading. Suspend your disbelief and imagine this is wonderful. The following tissue is the subject. The following tissue, I just clicked forward to the answer before I asked the question. I don't need your laugh from the kitchen. God! The following tissue is subject to conscious control. A, skeletal muscle. B, cardiac muscle. D, I am failing, which is a D, as I just said. A cut. Okay. At this point, like, I usually give up in the lecture because it's in the stars and it's not supposed to happen. I cannot do it. This lecture has to go up tomorrow. Then I will not do it.